from different parts of the world. Thank you for your questions. Thank you for your excitement. It's really a pleasure to read your questions and comments and have our scientists here answer them.
Yeah, thank you very much. Stay tuned. We've got another dive in about, let's see, how many hours? About 10 hours from now, we're diving again, I think.
What time?
I've heard that rumor.
Yeah, Argus is still moving.
4 a.m., so it's the same crew.
Oh, 4 a.m.?
Yeah, 4 a.m.
Wow, we'll have a lot of blue water.
Blue water.
It's a lot of blue water, too. It's a deep one.
Nice.
Oh, wow. How
deep?
many?
Oh, 3,400.
Yep.
Oh. Whoa.
Wow.
Well,
right
if you're on
at
Hawaii
4.
time, don't get up at 4.
Make sure you dream a lot and then tell us your dreams during the blue water.
Right, a little bit.
That could be dangerous.
So close.
Yeah.
Was
I'm
it
not
naked?
leaving until I don't see the bottom anymore.
All
That's
right.
all there is to it. I ain't leaving.
Well, that's generally the way it works.
Okay.
You start going up, eventually you don't see the bottom.
We are laid back like
And again,
20, 45, 50 meters. Yeah. Do you want me to just hold position and just let it settle out? Okay, that's fine. We made our best effort. Bridge
Oh,
nav,
you got
hold
really
position.
close. I never thought you were even going to get this close. Thank you so much for trying.
No
Ooh.
worries.
And we're there. We're basically there, as Trevor said.
All right, well, let it settle out.
So before we come up, we are going to
right.
perform our private ceremony. Are we ready for it?
Not yet. The ship's still moving. Argus is still moving. So if we want to leave the vehicle sitting still for this ceremony, then we should wait a little while.
Oh, okay. What's a little while?
I will tell you once I know.
Oh, okay.
Couple minutes.
Okay. So to our viewers out there, thank you for your questions, and I love to hear your names. I love to say your names.
There's another one, sea lilies. Sea pens or sea lilies?
What's that?
Lower right.
Oh, the animal?
Yeah,
I'm not sure what that is.
not that one.
No, that's
That's a corallimorph,
a Paragorgia.
Paragorgia. Oh, or you can't see it anymore. Lower right.
Antonella, that was a great try. I really appreciate it.
Yeah. We're so close.
Yeah, but let's say we're there. I mean, we're pretty much there.
You can see on Argus sonar how it drops off on the far side. So
Yeah.
Argus sonar can see the entire summit. So I'd call that a win.
Yeah, it's a
You're
win.
the kind of guy I want to go on a hike with, Chris. We're there. It's fine.
Well,
count
even
till
not
you've
even the
summited.
regular camera looks like you're looking across the flat top
Totally.
there. So
Yeah, I think
Full
we're good
summit.
there. We're probably 30 meters from, is that about
25,
right?
yeah.
25-ish.
Yeah.
So
It's probably,
and if there are a lot of
Are we
animals
happy?
on the summit, we would
Okay.
see them
want to do the
already.
closing ceremony?
sure.
Yep.
Okay, cool.
Okay, so
ROV's
to our
ready.
viewers, at this time we will be going radio silent to conduct a private ceremonial protocol to acknowledge and honor the biocultural resources that are present and that we are interacting with and impacting. We will be back shortly.
Thank you all very much,
Thank you.
and we'll see you in 10 hours.
Roger.
Excuse me, I forgot. I'm going to go down to the lab and prep some chemicals. So I was sort of leaving temporary, and I was thinking, oh, I'm leaving. That's it. But I'll be back up. I'll be back up here in just a little bit for a while as we ascend. So.
Sure, yeah, sorry. Yep.
All right, well, we have to ascend 1,400 meters or so to the top. It'll probably take us about an hour and 20 minutes.
Yeah.
Yeah. And maybe we'll see some cool open ocean critters.
That makes sense. Nothing makes sense. There, okay.
So the next dive is not on this seamount. It's, does anybody know what the name of the next seamount is?
Oh my gosh, starts with an S, I think.
Sovereign.
Sovereign.
And with us is Dr. Beth Orcutt. Thank you for joining us. And
I love joining for the blue water.
Really? All right.
You can't detect the sarcasm in my voice?
No.
No, it's fine.
Well, we had some good questions about general questions about like the manganese that settles out or comes up through the pores, and where does that come from? Where did all the minerals that are in the water come from? How does it get there? And is it in higher concentrations in different oceanic areas, or is it kind of uniform throughout? Tried to take a crack at answering that a little bit, but
What was your answer?
Well, saying the encrusting process is a lot more dependent upon how much additional sediment is in the water column. So that process, the manganese encrusting process, happens everywhere in the open ocean at that depth, around these depths, but that if there's more settlement of other sediment, then it covers over those encrusts and not able to, or if there's stronger current, or if it's not stable environment, then it can't settle out. It needs basically a stable, low sediment environment to allow that process to happen, because it happens so slowly.
Mm-hmm. Yeah, that's true. Yeah, and I'm not an expert in manganese geochemistry. But essentially the manganese in dissolved form in the seawater, or as well as remobilized in the sediment pore waters that comes up from below. The manganese sources from a variety of places. It could come from hydrothermal inputs, also terrestrial inputs of manganese to the systems. But actually, I don't know whether one of those sources is more dominant than the other. I think we're only just realizing how far dissolved metals travel from hydrothermal inputs.
Huh.
Originally, it was thought that most of that metal, and it is true that most of the metal does precipitate out very quickly, but there is a sizable fraction that can travel tens, hundreds, thousands of kilometers away from hydrothermal inputs.
Oh, wow. Oh.
Because that metal gets bound by organic ligands, and it basically protects it from being oxidized and precipitating out. Those ligands are formed by microbes. There are several researchers that are trying to better understand that process of how these ligands and siderophores and other organic compounds are generated by microbes near hydrothermal systems, because it seems that that process kicks off very quickly as the water comes into contact with seawater. And you know, what is the mechanism for that? Is it kind of the microbes want to use it right away, but some of it kind of is a loss? So there are various studies trying to understand those sources, where they're coming from, where they're transported, and then how that plays in with what we see in these mineral crusts. But these mineral crusts precipitate over tens of thousands, hundreds of thousands, millions of years.
Yeah.
So even though the concentrations of these metals in the water is relatively low, over time, it builds up.
Yeah. Cool. I didn't realize a lot of that came from the hydrothermal vents.
Well, again, I don't know what the relative ratio of contribution is, but it is a source.
Yeah. Yeah, I mean, the oceans are a giant solution source of many different chemicals and materials. I mean, that's where the corals and these other kind of, calcium carbonate forming organisms, they get their materials out of the ocean and are able to aggregate it. I mean, that's one of the reasons why we're concerned with acidity of the oceans, because of the acidity, as the oceans become more acidic, it becomes harder for these organisms to build their calcium carbonate skeletons.
Correct.
For the availability of their aragonite and things like that. And
right.
Yeah, and might even affect these processes that you're looking at if over at some point the acidity changes at depth.
Yeah, I don't know if anybody's studying that, what the potential ocean acidifications might be in the deep sea related to these processes.
Mm-hmm. Yeah, but a lot of these waters that we're studying or looking at depth are older waters, right? I mean, they're because of that thermohaline circulation. The Central Pacific waters are, as far as I know, some of the oldest waters on the planet.
Yeah, that's true. They've circulated all around before getting out here. And we're probably intersecting different bodies of deep sea water. It's not all one water mass moving out here in the Pacific.
Yeah, yeah. Well, it's a gigantic, Pacific is the largest ocean on the planet, so it's it's got a lot of different sources.
Yeah, well, that's always a fun thing to think about, whether there's one ocean or multiple oceans, right? Because technically they're all connected.
Yeah.
But yeah.
I mean, I like to think of it as one ocean. I like that concept.
Okay. Yeah.
Different geographic bodies.
Right. Yeah, I like seeing some of the different map projections. How we view the world is so dependent on how we interpret maps.
Uh-huh.
And I've seen some really great maps that put an ocean-centric view forward, where the land masses are divided up to really show that connectivity and that it is one big body of water with some land masses in the middle of it.
And a different perspective of people. We have Polynesians who, for before Western travelers were exploring the world, they had navigated this largest ocean and don't view the ocean as disconnecting land masses. They view it as the highway connecting places, and the oceans are part of their perception of the universe. I mean, it's not like a terrestrial-centric focus. It's more like this oceanic focus, and water as highways between areas and things like that. That was part of the World Heritage designation for Papahanaumokuakea was considering that perspective of the Hawaiian people and early Polynesians that came to Hawaii as movements upon the sea.
Yeah.
This kind of migration and movement around the oceans is part of a World Heritage. What's the word? It's I forget the categories of the World Heritage.
But there's like biological and cultural.
Yeah, but they're outstanding universal value. So it's one of the criteria of the World Heritage, and outstanding universal value might be something like that movements upon the sea, and that was one of the criteria for designating Papahanaumokuakea on the cultural side as a World Heritage Site, because we're a natural and cultural feature. But this cultural seascape concept was pretty new. I mean, there hadn't been a lot of designations, World Heritage designations in the Pacific. And so we tried to change some, oh, what was that? It's a weird little critter in Argus view. That.
Oh, that's, oh.
I don't know what that is. It looked pretty sizable. Yeah, so, you know, it's a different, the perspective from oceanic peoples is very different from terrestrial focused people that live in continental or adjacent or along continents and move along the continental shelf.
Yeah, I forget now who this quote is attributed to, but essentially, like, you know, it boils down to, like, it's so weird that we call this planet Earth.
Yeah.
It should be planet Ocean, right? And why did we pick the feature that was only 30% of the surface? And that's...
Yeah, and that's what's, I think that's, and you know, coming from the educational perspective, that's the hard thing to, it's a hard thing to bite into because people are, you know, they're associated with terrestrial areas, they're comfortable in terrestrial areas. I mean, I mentioned this in another blue water dive, but you know, even in Hawaii, pretty large percentage of our population does not go in the ocean, doesn't know how to swim even. And you would think
that's probably
that,
true for most coastal communities.
Yeah, you would think that, at least in Hawaii or ocean, you know, and these low-lying islands, that's also true. Like in the Phoenix Islands, a large percentage of the population doesn't go above their knees into the water, you know, and they're not like they're all fish. So I mean, we're just so focused in general around this terrestrial perspective that to do things like we're doing aboard Nautilus and some of these other things, it's hard for people to wrap their minds around, I think, this oceanic perspective, this water perspective, and what's beneath, because you can see the surface of the ocean and everything looks fine. And, you know, beneath the ocean surface, there might be a lot going on. You got the ocean acidification, you got debris issues, you've got all these other overfishing and other impacts. But people in general, I think, don't think from a water perspective, you know, and that's one of the things that I think we need to change around to really, really take care of our planet better as oceanic planet, you know.
So I hear your shift tomorrow also has some blue water coming up.
Some big blue water, it sounds like.
At 4 a.m.
Yes.
We're gonna have to come up with some topics for.
Yeah, I say
3,000 meters of blue water tomorrow at 4 a.m.
Consult your pillow.
That's when I'm definitely at the top of my game at 4 a.m.
Right. Yeah, it's when you're most inventive.
Yeah, because we're going deep. 34. What'd you say? 3,400 meters.
Something like that. Yeah.
That's the deepest dive.
I think so. I believe that's going to be the deepest dive of this expedition.
Well, I'm very excited to see what's in that depth. So.
Yeah, hopefully there's some animals.
There's definitely going to be some rocks.
There will definitely be rocks.
We can pretty much confirm that.
Yeah.
And sediment.
Yeah.
So what's the push cores, did they all kind of have, so the push cores, for those who are just joining us, are the kind of like this tube that we shove into sediment and collect sediment samples, and the length of that is like 10 inches or so?
It's more like 12.
12 inches, okay, so a tube that collects a sediment sample up to 12 inches, and we've collected several of those on this expedition. And did you looked at that in the lab? Like, are they all similar, like granularity, sandy kind of? How would you describe them?
Yeah, they're a very coarse sand material. So you can see individual grains very easily with the eye. You know, 1 millimeter, 2 millimeters, very angular fragments. Although there's some rounded 4M tests, but also like broken carbonate, broken debris.
Yeah.
Some black flecks in there, probably of crushed up mineral crusts. There's not a lot of layering in the cores that we've collected, as opposed to, for instance, if you were coring in a very clay-rich continental shelf setting, you would see like different layers in the sediment reflecting different chemical processes that are occurring. So it'll go from oxic to oxic conditions and all the oxygen's gone, and then microbes are starting to use other substrates to breathe, and then it maybe gets anoxic. And you can visually see that the color of the sediment is different. We don't have that here.
Yeah.
It's just sand, light colored sand all the way down. It's actually been very difficult to get sediment push cores here because of that.
Yeah.
That there's not a lot of cohesion to the
stick.
material to keep it to stick in the tube. And
You just got to
also-
be fast.
Yeah,
Trevor's
that's true.
mastered the art.
Yeah, so we've gotten 2 cores successfully, which is enough to see what that material looks like as a reference point for the other samples we're collecting.
Yeah. So a lot of that material is also like that marine snow, right? Too, like kind of
Well,
calcium
there may
carbonate.
be some in there, but like your true marine snow of like organic material, there's not a lot of that.
No. Huh. So it's
Doesn't
all
look
kind of
like it.
generated on the seamount or in the adjacent
Yeah, it looks
area
kind
of
of
the
like
seamount.
debris, regional debris, right? So
Okay.
maybe if there's, because it's so angular, that's what gives me that thought that it's, yeah, coming off, it's depositing from material that's coming off of the seamount.
Oh, interesting. Yeah, that's very different than the shallow water, like the sand that we have around the shores of Hawaii, which is mostly coral derived, and like critters like parrotfish, they chew on the coral and then
Right.
they poop it out, and that's the sand. You're walking on parrotfish poop when you're walking on Hawaiian sand. You know, we don't have continental granitic inputs or other things like that, so you're looking primarily at that when it's white, when it's pure white, it's calcium carbonate sand, or
You've got the nice black
black
and
sand,
green sand beaches.
yeah, even the olivine green sand beaches, and some are even the red from the cinder cones. Those are my favorite in Hawaii.
I haven't seen red.
Oh, it's
I've
beautiful.
seen black and green.
It's just like rust red.
Yeah.
Those are typically associated with secondary eruption from a volcano, and it's ash cinder from those secondary eruptions, and it's red.
Okay.
Yeah, so we have green, black.
Where are the red sand beaches?
There's several. My favorite one's on Maui, like out by Hana on Maui, but there are a few around. I'm trying to think if there's one on Oahu that I can't think of one on Oahu, but Hanama Bay has a little bit of that. Hanama Bay was a secondary cinder cone eruption, but the best one in my mind is that, yeah, it's off of near Hana, on Maui, and yeah, it's really if you Google that, red sand beach on Maui, it's one of the most spectacular places I've been for snorkeling and stuff. It's really cool because it forms that cinder ash conglomerate.
Okay.
Yeah, it's a very different composition of the stuff we see down here from shallow water habitats, because certainly, I mean, it could be coralline. Some of it could be coralline, because if these were atolls, if these were geo, you know, where it was at the surface and that eroded material from calcium carbonate animals had kind of drifted down off the edge of the seamount, right?
Or they're still in that flat top geo.
Yeah.
You have breakage events.
Mm-hmm. Yeah, it's really neat. I mean, some of these areas, they had quite a bit of sediment, quite a bit of that white, fine sediment. Not very deep, though. That was the problem with the cores, right? I mean, it went down maybe 2 inches and hit bedrock, hit the basalt or calcium carbonate bottom. Yeah, but that area we saw yesterday, that was super cool with those like really blocky, angular formations at the top of that seamount, that geo. That was super cool. Didn't expect that.
Yeah, blocks of carbonate that have been crusted over with manganese.
So how do you preserve the bacteria? So you take the rocks, you collect the rocks, the squishy rocks is the ones you like to have a little more crunchiness.
Well, we've gotten all kinds on this
Yeah.
cruise. So ones that are highly altered and others that are only moderately altered.
Yeah. You use your chisel and hammer and you bang off the crust on the outside, and then you preserve it in some type of material to preserve the bacteria and microbes?
Yeah, so I'm, for the rock samples that we collect, I preserve sub-samples from each mineral crust. So in some cases, I'm just freezing it directly to preserve the genomic material that back in the laboratory we'll use chemical techniques to extract the DNA and nucleic acids for genome sequencing. In other cases, we're preserving them in a buffer to maintain the stability of a molecule that we're looking for. Sometimes we use a different preservative that's kind of like an antifreeze, so that when we then freeze the samples, it keeps the cells from rupturing.
Oh, okay.
So that we can do analyses where we want to maintain the intact cells and like count them, or do microscopy, do flow cytometry. And then some of the samples are used for doing some potential rate measurements. So put the rocks in some sterile seawater, give them a little bit of food, and measure changes in chemical concentrations over time.
Oh, okay.
So from every given rock that we pick up on the seafloor, I probably generate about 15 different subsamples.
Oh, wow. That's wild. And so for the bacterial samples, I mean, how good is, so let's say you extract your material, you've isolated the well, you've even isolated the bacteria, but you isolated the DNA sequence out of those, and then try to compare that against a database of known bacterial DNA structures to see if you can find matches?
Yeah, that's essentially what we do. So there are public databases of genome sequences from all over the world. One technique we'll use is to try to sequence a specific gene that all bacterial and archaeal cells have, which is it's what's called a taxonomic marker gene. And we'll basically use that as a survey of the microbial community in every given sample to get, let's say, hundreds to thousands of copies of that gene from the different organisms that have been in the sample. And then we'll compare those sequences to this reference database to get an idea of where they fall in the tree of life.
Yeah.
For some samples, we may do deeper genomic sequencing, where we actually try to sequence all of the genomic material, not just the taxonomic marker gene, using techniques like metagenomics, or even single-cell genomics if we have samples that are really interesting. But they basically are the same concept, where you're sequencing the DNA and then comparing that to established archives, where especially references against cultured isolates, where we kind of have a really good idea of what all the enzymes are and genes, what kind of proteins they make. And so we'll compare our sequences to those, see how well they match, make interpretations of what we think those organisms might be doing. So, yeah.
Huh. That's cool. And so, like, your best wild guess of the samples that you collect, I mean, do you expect to find a lot of, do you expect to find a fair amount of new bacteria that that aren't in those databases, that hadn't been sequenced before, because this area hasn't really been collected from or explored? Or
this is an interesting concept, because it gets at the heart of one of the conundrums of microbial ecology, is how do we define what a microbial species is? So we have I have a general idea of what families, so I expect to find. So if you think back to your basic biology class, you may have learned the 5 kingdoms, maybe you learned the 3 domains of life, right? And that you have all these different taxonomic levels, like phylum, kingdom,
right.
Order, family, class, genus, all that kinds of things. And so we might not be able to predict down to the species level what we're going to find, but I have a general idea of the families that we'll find. if we were to just focus in at the species level, like, is this DNA sequence, 99% similar or less to known things, then we would definitely be finding lots of new things. But the way that microbial species evolve, the rate at which they pick up genetic mutations is much faster than in other domains of life. And so a lot of times those definitions are kind of squishy for applying to microbes. So yes, we'll find new things, but we'll also be finding they're like just variations on a theme. And they probably will have a lot of the same functional capabilities as their closest relatives.
Yeah.
Yeah, so it's not a black and white answer to your question.
Yeah, it never is, really.
No.
The deeper you go, the less black and white it gets most of the time.
Yeah, yeah. So we, one of the things we've been doing in my group is trying to gather together representative samples from the deep sea where people have looked at the composition of microbial communities on different types of rocks, on manganese nodules and sediments, et cetera, to have kind of a good reference library so that when we get the DNA from these new samples, we'll have an idea of, do they look more like fresh rocks? Do they look more like old rocks? Do they look like rocks from oxygen poor areas or oxygen rich areas?
Yeah.
Can we say anything about, these samples have higher iron content and they have higher relative proportions of particular groups of organisms? Does that mean those groups of organisms are involved in cycling of iron? Those are the kinds of questions we'll be looking at.
And so given that most of the processes for animals, at least at this cold, extremely cold, dark depths, are very slow, do you see a similar slowdown in like mutations of bacteria and stuff at these depths, or are the processes completely different where they're mutating and adapting and changing, or is it similarly slow?
I think it depends on the group. So there are some data out there about particular groups of microbes that are found in the deep sea as well as in the deep continental subsurface that seem to have incredibly slow mutation rates for their genomes. Just like mind-bogglingly slow. But then there's other clades that seem to have a lot more diversity, genomic diversity. And so there's not like one answer to those questions. And we don't really know why that is. What is it about some groups? What is the evolutionary strategy or adaptive strategy for those differences? Yeah, and then in the sediments, it hasn't really been studied so much in the rocks, but in the sediment realm, there's definitely a growing body of knowledge about there are some groups of organisms that, rain down in marine snow, and they are particularly well adapted to survive in the kind of one-way ticket down in sediment to actually grow. But they're growing very, very slowly. Whereas other organisms kind of are just like slowly dying. So again, it's kind of, what is the unique capability that those microbes have to actually prefer that environment? Yeah,
that's pretty cool. So what drove you to get into microbes? Why microbes?
Yeah, I don't know. When I was a freshman student in college, I had gone to college because I found chemistry interesting. And so I was like, maybe I'll try that. I don't really know what I'm doing. And I had a chance to do an internship in a marine chemistry lab, wherein was kind of my first exposure to environmental science and the recognition that microbes are responsible for a lot of the chemistry that occurs in environments. And I found that fascinating. And so I decided to kind of change the direction of my education to kind of focus on that. So yeah, it was really just more the fascination of realizing that this microscopic forms of life had so much power to really shape the environments, both kind of naturally and responding to anthropogenic change, bioremediation, those kinds of things. So I guess that's why. I don't know. I didn't really have a lot of exposure to it before college, but it just seemed like that's something that was pretty cool.
And it seems like the tract of the deep sea microbe is even more of a sharp turn in the field. I mean, microbes drive everything, right? I mean, there's so much research and so much fascination now about how microbes control even our human beings' emotional state of the microbial community in our stomachs, right? In our guts, and how that even affects, like I said, our emotional state, or how it even controls animals in some cases. I mean, some biological behavioral modifications, you know, but there are microbes in the atmosphere. We're looking at microbes on, you know, trying to search for microbes on Mars, you know, but why the deep sea microbes? What?
Again, because of the exposure in that laboratory, that was kind of the environment of focus, was continental shelf environments, and I just kept going deeper and deeper. You mentioned the interest in looking for microbes on Mars. That's something that I'm also interested in, this idea of like trying to understand how microbes survive and what could be considered challenging environments on Earth, even though they're like some of the most abundant environments on Earth. So it's very much from a skewed human perspective. Yeah, so trying to understand like, how does life eke out a living when there's not a lot of energy? And then what would be the signature for that, so that you would know what you could maybe look for on other planets? Is part of the research that my team does as well. So that's why.
Do you work on like stratospheric microbes too? And
no, just deep sea.
Just deep sea, yeah. I mean, would these be considered like the primordial microbes in a lot of ways? I mean, some of the oldest on the planet? Oldest groups,
depends on the environment you're in, what kind of microbes are the dominant groups. If we look at the tree of life as our guide for how organisms are related to one another, and you look down at the deepest branches of those trees, the organisms that reflect those deepest branches often have a hydrothermal environmental signature. So they're organisms that are adapted to living in like anaerobic environments, high temperature environments, environments without a lot of oxygen. And so based on those kinds of inferences, the working hypothesis right now is that life, the last common ancestors for life on Earth probably had a hydrothermal origin. So there's a lot of research into that in terms of what is it about hydrothermal conditions that allow, you know, catalysis, which basically can then turn into enzymatic pathways. How can organics form from inorganic compounds under hydrothermal conditions, to kind of chase that hypothesis. So the organisms that are likely dominant here are in parts of the tree of life that are like much further radiated away from those deep branches. And so what we don't have is a good like molecular clock for our tree of life. So even though these organisms probably don't reflect like the last common ancestors, they still have probably been around for a really long time.
Yeah.
Yeah,
it's pretty wild to think a lot of these microbes were responsible for our atmosphere on the planet. I mean, we wouldn't have an atmosphere if it weren't for a lot of those early chemosynthetic or sulfur vent kind of microbial activities, creating, you know, the basis for life really on the planet.
Right. And then, you know, what was it about the organisms that could then springboard from that to be like, oh, I can take advantage of even yummier food, but I'll make oxygen as a byproduct.
Right.
And then that's responsible for the atmosphere we have now.
Yeah, that's crazy. I mean, it's just crazy to think. that creating that whole process of creating habitable Earth.
Yep.
Yeah.
Yeah, and so that's part of the question for some of this, what's called exobiology research of like, are there environments that could be habitable for different types of metabolisms, even though it might not be habitable for humans, could be habitable for other forms of life.
Yeah.
It's not, it doesn't have to necessarily be that, you know, all roads lead to generation of oxygen and conditions like what are on Earth.
Right. I mean, had they, so I mean, now we're looking at actually harvesting some material off of asteroids, right? And before in space, I mean, do you expect we'll see some things like microbes on some of those, some asteroids or things like that? Meteorites?
I don't, well, yeah, so there's a whole other body of research about whether life on Earth is actually not originated here, but was originated somewhere else and was seeded on Earth, this idea of panspermia. And so that relies on the ability for asteroids and comets to be able to transport things like organics and cells, which is not bulletproof proven. And it's a very hard thing to look at in terms of how do you know this signature you're looking at actually came from the asteroid and not from the sampling equipment you brought from Earth?
Oh, right.
So that's a whole nother area of research within exobiology, is this idea of like, you know, false positives and like really convincing that what you think is a signature is a real signature.
Yeah, yeah, that must be very challenging. Try to figure that out.
Yep.
So your lab's on the East Coast, right?
Yep, Bigelow Laboratory is based in Maine, on the Gulf of Maine.
Oh, nice.
Yeah, and I think it was snowing there, so I'm gonna have a really hard right turn from my flip-flops out here.
That's a hard shift, definitely, for sure.
Yeah. But it'll also be nice and Christmassy.
That's true. That's what I definitely missed in Hawaii for sure. Is it still doesn't feel like Christmas every year it rolls around, just because that change in the weather and the snow and all that.
Yeah, I'm interested to see what it'll be like when we return to port soon, and what the Christmas feel, the holiday feel is like on the island.
Yeah, it's there. It's, you know, it's a good holiday in Hawaii.
So one of our viewers was asking earlier if there are enough scientists to process the samples that we are collecting. And of course we have different types of samples, right? We have the geological samples, the biological samples, and of course the microbiological samples.
Yeah, so there's a couple different ways I would answer that question. The Ocean Exploration Trust is actually a kind of unique model for how oceanographic research is done, in that it's kind of almost like a service provider, in that there's kind of set samples like rocks and animals, et cetera, that are collected and then sent to repositories so that scientists from all over could request them without actually having to come on the ship. And they provide ways for shore-based experts to actually be part of the expedition. So in addition to the chat we have on the Nautilus Live page, where folks are sending in these questions, there's like an additional like science channel for the shipboard scientists and shore-based scientists to communicate. And that's kind of unique. And that's partially a reflection of that the focus of the staffing on these cruises is more for the core of exploration, having the science fellows, having a lot of technical staff, and so there's not a ton of berths for scientists to come aboard. Whereas other vessels that scientists typically use, you can have a much larger science party, but you don't have all the technical support that the Nautilus has. And so depends on what you're trying to do. So I had to be mindful of that when designing my research plan for this cruise. Hey, to be mindful of like, okay, it's just going to be me out there collecting and trying to preserve all these samples. So what is actually realistic? How much can I get done per dive? So whereas on other expeditions, I might have requested more samples because there'd be more people to help, I came up with a strategy that was feasible for one person to accomplish, or one person with help. But knowing that there's all kinds of other scientists on shore who are going to be part of helping analyze the samples. And with that, I'm going to turn it back over. Yeah, no worries. It was nice hopping into this.
Yeah, thanks, Beth.
This shift, and I guess we're back in the water at 8 a.m.? No, we're back in the water at 4 a.m. So I'll be back on at 8 a.m. Hawaii time.
Thanks for joining
Tune
our
in.
watch.
Yeah.
Thanks.
Okay, thank you.
Bye.
Chris is back with us after doing some of his
What were you doing down there?
sponge labus. He's processing his sponge samples. We're getting ready to. It is.
Were you preparing the couches for the sponges? Were you preparing the couches for the sponges? Oh.
Chris, you're off SPL.
Oh, connect yourself. So
Chris,
you were.
Could you hear me in the wet lab?
No.
That system must be muted.
Well, I had the fan on because I was preparing formalin for the anemones and the other things. That fan's pretty loud. So
you didn't just mute me?
No, I did not mute you at all. I could hear nothing but this whirring motor. So, and then I was also up in my room. Took the opportunity to prepare the sponge pieces that I'll be taking with me and get them out of the lab there and get them out of everybody's hair. So I just have small pieces of each of the sponges, and they're just completing their drying in my room. I've got a nice little lamp drying them right now on the desk.
You have a lab in your room too?
I have a lab in my basement, but it's not like a big lab. It's a
in your
Oh,
house.
yeah, I knew you had at your house, but in your room here?
Well, I've made my desk into a little mini lab. Here
Chris sleeps with sponges. Come on.
Yeah, that's what I found out today.
John, my roommate, is a little concerned about the smell, but there is no smell. But there isn't. They're dry enough. I don't bring them up until they're not smelly.
Well, you don't know.
No, I know. I sniff. I use a sniff test on all of them.
So you're preparing the formaldehyde, you said?
Yeah.
Ooh, that was
So
a bigger fish.
just helping Steve out a little bit with that. And I'll be down there in the lab afterward, helping him process the specimens that came up.
Yeah, what did we collect on this dive? We had a couple anemones. We had that beautiful black coral with the pink squat lobster.
Oh, that's right about that one. We have
what else do we have?
The serianthid, the big, huge serianthid weird thing.
And the crab.
And the crab. That's right. And there's something else. I'm missing something.
Anemones.
The big starfish.
Oh, yeah.
Yeah.
There's a lot of stuff that potentially could have escaped.
Yeah. So starfish do not go into formaldehyde because formaldehyde ruins their ossicles for morphological examination. But I assume that Steve is probably going to snip some tube feet for DNA analysis and get it into ethanol. And then the anemones will go into formaldehyde and ethanol because we need both for morphology and for genetics. Same thing for the serianthid. And what else? The coral will be all alcohol, and the crab will be all alcohol. That's the plan.
So how long is it going to take to prepare all those samples?
Well, I kind of bail when they get to the rocks, so I leave them after the biologics, but it usually takes 30 minutes to an hour or so. And then Steve and Kainala, how long do you normally stay with the water and the rocks? Are you there 2 hours maybe, or?
Yeah, it depends how much we have. Usually like an hour and a half.
Hour and a half?
Yeah, not too bad.
Yeah.
But I think this one, there's not too many, so it'll probably be like an hour.
Yeah.
Yeah, I don't think there's any water samples actually.
Oh, you're right. That's right.
Yeah.
Wasn't really anything to sample the water above.
Yeah.
Okay, well that'll go faster.
Yeah.
Particularly since we got to be back up here at 4 in the morning.
Yeah. So it kind of works out.
Yeah.
So what was the driver for choosing a deep dive on this next one? Next seamount?
It's a deep seamount. Is one that we needed rocks from anyway, and there just wasn't any shallow. It's one of these conical ones that never came.
Oh, so the top's very deep as well.
Yeah, and then the ridge itself was deeper. That looked like it was a good place to dive. So, yeah, I suppose we could have dove I'd have to take a look at that, but I know it was deeper in general.
Uh-huh.
And also kind of mixing them up. We had 2 dives that were 3,000 meters, and this would be a 3rd dive out of 14, so that's not bad.
Yeah.
You'll see some different critters down there. It'll be sparse, but there'll presumably be a few interesting things that we haven't been getting.
Cool.
So which seamount is that?
It's Sovereign Seamount, so right up there.
Oh, okay.
Yep.
Sovereign Seamount.
Yep.
Did we find the name origin for that one? Is another
another
ship?
Another ship, I'm sure.
A lot of these things are named after ships. This one that we're on right now is named after, not this one. This one, the previous one.
Hey, how many dives have we done on this cruise?
This is the 10th, I believe.
10th, okay.
Yeah.
Cool.
It's actually working out okay with it. This storm just sort of holds off. I think we'll get the 14 dives, which is what we were shooting for.
Nice, awesome.
So video, Aaron, questions for you. So what other habitats are you going to for your video projects? You got the deep sea, and what else are you working on?
Video Erin's out of the house.
Oh,
at the moment.
I didn't notice you.
Yeah, I think she just ran downstairs quick.
Oh.
Sorry.
I didn't see. Sorry.
You picked the wrong question, Andy.
I did. I blew it. I was on a roll there for a while. You missed it, Chris. How about we get some commentary from Kainalu? So what do you think, man? This is like your first deep sea cruise, doing science, doing the logging of all these critters out here. What are some of your thoughts at halfway through the cruise here?
I think I'm starting to get the hang of it now with the data log.
Yeah.
Yeah, it's like definitely a lot of multitasking when there's a lot of samples and stuff we're seeing. So kind of finding the groove for that, I guess. Trying to keep track of everything. And
it's a lot of work, isn't it?
Yeah, learning a lot though. So it's really, yeah, it's really fun, I guess.
Yeah.
Yeah.
So you got an interface here where you're typing in some notes about things we're seeing and
yeah, so you usually try to
species.
Catch the Oh, yeah. Thank you.
Erin's back, by the way, if you want to pop that question at her again.
Oh.
She's
Uh-oh, what was the question?
Oh, it's a real toughie.
I asked what your favorite color was. No,
Oh, I got that one.
I was saying, so now you're working on you're doing this today now with the video project in the deep sea, but what other environments and habitats is your passion for videography taking you to? What other projects are you working on?
In my other life, I film wildlife all over, so I do a lot of long lens work, so that's kind of, you're quite a distance sometimes, or you're in hides or in a car, if it's in like an Africa shoot, and you're filming a lot of behavioral stuff, so my favorite thing is the long shoots that you're sitting and waiting for something to happen. So my favorite animal to film are brown bears, and I've done quite a few brown bear shoots.
What's the animal?
Brown bears.
Oh, brown bears.
Yeah.
Okay.
In Alaska and British Columbia. And
yeah, so I do mostly Alaska for it. And I've done a lot in like Katmai National Park and that kind of stuff. But yeah, that's my favorite. But I get to go all over for it, so it's pretty fun.
Which one won the Fat Bear contest this year? I didn't follow that.
That was, oh no, I don't know the answer. I watched it too.
If you don't know, if folks who don't know, there's I can't remember the cam, but it's kind of a contest of which bear is like successfully packs on the most pounds before hibernation and catches the most fish.
Yeah, it's the explore.org Katmai Brooks Falls cams.
Yeah.
And it was kind of a way to like raise awareness on the importance of fish in the bear ecosystems, and it turned into this really popular thing to bet on and like get into.
Yeah, it was really, really cool.
But yeah, so that's my other job is just filming wildlife.
Neat. You got any good bear stories? Bear scary stories?
Let's see. I've had lots of close encounters, but always pretty safe. We normally have guides with us just because when you're filming, you tend to not pay attention to what's going on around you. I do go out and film on my own quite a bit in Alaska, and especially when I got started.
Yeah.
And I did have some close ones. I've had some bluff charges where they false charge you, and then they pull back last minute. But yeah, they were all situations where I had bear spray, I had the little blowhorn thing, and I had a flare, so I was prepared for those. So I tend to be quite safe when I go in. But yeah, I've never had, I think the worst encounters I've had have actually been with people.
Yeah.
So bear spray can be used for more than bears. So I've never had to, but yeah, I'd say the people encounters have been scarier than any of my animal encounters.
That's pretty cool. I mean, it's quite diverse from the deep sea to bears in Alaska.
Well, Steve's the same way. So the other video engineer, he does a lot of like macro camera work. So he'll do a lot of like tiny insects or amphibians and reptiles, like really small ones. So he does a whole different type of filming as well.
It's like the folks at home might not know, but when we zoom in on critters, Aaron's like over there just making sure we get the best image of them. Do you have to adjust the aperture and all that stuff on the lens, or is there no aperture, or how does that work?
Yeah, so we have aperture that we're adjusting, and then we have focus and zoom, and then like the blacks and stuff like that. The camera tilt and pan is actually controlled by the ROV pilots, so it's a lot of working together, and a lot of times it's the ROV pilots' moves that make the shot look really good. So yeah, it's kind of a team thing.
And sometimes I make them look really bad.
Jiggle and shake too much.
But it is quite fun because the ROV's never completely stationary unless it's like landed. So you're doing a lot of practice on focusing in and out, because any little movement, if your iris is quite open, then you get quite a bit of change in the focus area.
Yeah.
So it's good practice for my other jobs is being able to do the focus while you're moving.
Yeah, and you become probably a master at depth of field, because of a lot of these organisms have such a, you're focused in and they're so wide, right? I mean,
yeah.
and the focal point's not clear.
No, it's some of those sponges, or not even sponges, the corals tend to be the worst. Those ones that even when it's in focus, it doesn't quite look in focus because it's just so much going on, and it's almost like a translucent looking. Yeah, it's quite fun. It's a fun challenge.
Yeah, especially how do you figure that out with the aperture? I mean, that must be.
We have